Here we go. We are recording. This is a PCR lab on a PCR gel for our PCR samples that we did last week on our recombinant DNA from Landoltia punctata. Here we have our agarose gel. It's roughly 1% agarose gel. I'm going to put some ethidium bromide in this gel. I'm going to put about 6 microliters in. Now, ethidium bromide is a known mutagen. Check how blood red it looks. You know what? Let's put 12 in. Let's get crazy. Let's put 12 in. This stuff is a known mutagen. That's why I'm definitely wearing gloves. It actually gets in between your DNA base pairs. And it is known as a result to be a carcinogen, a known mutagen. If molecules get between your DNA base pairs, your DNA replicates differently than it normally should. And who needs that? The answer is no one. So this is a known mutagen, ethidium bromide. Ergo, I'm wearing gloves. Now let's pour our gels. When I pour these, there's a technique we use, and I pour them at the edge of the table. Here we go. Hello. So, let's see, I'm gonna bring this up to the camera so you guys can see. Swirl my gel, 1%. I cooked it this morning. Agarose gel, electrophoretic spec. I'm gonna pour these nice and deep. And when I pour pretty forcefully. I want to fill up both of these with a lot of agarose because I want a lot of gel, a nice thick gel. Done with that. Let's show you guys. So I poured it into these chambers, and I don't know if you can see the profile view, but it's really, there you go. You can see the profile view here, I think. It's really thick, probably a solid centimeter thick. And these little combs, these little plastic white combs, are purposely in the gel to form wells. Now, I chose 2 combs of 2 different sizes. This one is for period 1. That's you guys. Period 1. It says period 1 PCR, and there's going to be 8 lanes. Now, I chose 8 lanes because we have 6 tables, and I'm going to put 2 markers, 2 DNA ladders. And for period 3, I chose 10 lanes for 8 tables and 2 markers. Now, there's only 6 tables, but for period 3, we have 8 samples. I just did an extra 2. So to reiterate, this is PCR for period 1. M is for marker, T is for table, PCR, the date. M is for marker. Because I'm not doing this for every single student, I'm doing it per table, I figured we use 2 markers, 2 DNA ladders, right? So let's use one on the left and one on the right. On your left, I'm going to use the Rutgers ladder. This is the 10 kilobase pair ladder. And over here, I'm going to use a 5 kilobase pair ladder. I'll show you these in a minute. Oh, also, why am I writing it on paper? Because when we're done, we're going to take a picture of this, of the gel, and we're going to see the DNA light up in the gel. The DNA lights up because of the lithium bromide. The lithium bromide, when excited with specific UV light, it's actually UVC light. I'm fairly certain it's UVC light. I could be wrong. I'm pretty sure it's a wavelength near UVC. It's definitely UV. When it excites, it glows a pretty orange. And when it glows that pretty orange, we take a picture of it. We could see the DNA in the gel because of the lithium bromide. So the lithium bromide is really, really dangerous, but it's really, really useful. It's soaked into the gel. As the DNA moves through the gel, it's going to pick up the lithium and light up when we take a picture of it. We're not going to actually see the DNA move in the gel because DNA is transparent. It's invisible for the most part. It's really more translucent, I guess. So it's not very visually obvious. But if you do excite it with UVC light, it will glow orange. All right. Let's get our DNA samples ready while these gels cool in the corner here. So I have some loading dye here. I have my pipetter here. I'm going to dial my pipetter to 2.5. Zero 0.25. All right, let me get the PCR rack. PCR rack. And here is period 1. This is you guys. I took these out of the thermocycler on Thursday, I think it was. Period 1, 6 tables. I'll put these in. Period 1 through T6. Let me take off the tape. I have to be careful to leave these in order because I don't want to get them out of order. Even though I did label them, sometimes the tape pulls the labels off. But actually, it didn't really do it at this time, which is good. Depends on the temperature of the label and the tape quality. But I think it worked this time. So nevertheless, these are tiny tubes, hard to see. I have to make sure that I know that this is T1. Hold on. That's T1, T2, T3, T4, T5, T6. They're actually labeled here somewhat. You can kind of see them. Let's do the same thing with period 9. 9 back here. I'm going to put 9 in the back and pull off 9's tape. Eva, how's this coming through? Okay, can you see it through the screen? Oh, you're not? Okay, but you can see? If you want to, you can see no problem? All right, Viva can see, we're good. Viva's mask is like a gingham red. I like it, it matches this tape. Mine's a boring gray, but that's okay. My name's Jay. Oh, come on. Ouchie. Okay, whoa. Now these are backwards. So let me rotate this. Actually, no. Okay, because I want them to go in your order. That's 1, 2, 3, 4, 5, 6. And this is going 1, 2, 3, 4, 5, 6, 7, 8. Okay, so I did stagger them. This is 1, 2, 3, 4, 5, 6, and this is 1, 2, 3, 4, 5, 6, 7, 8 for period 9. Period 9 in the back, period 1 in the front. Here we go. Let's add our loading die. Now, I'm going to do something here that's a little unorthodox, just for in terms of time. I'm going to use one tip and add the loading die to all of them at once. You really shouldn't do this because it's easy to cross-contam. But this is a dead-end lab. I mean, all the important DNA is still in our cells in the freezer. So if I mess this up, which I don't think I will, it's not going to really matter. It'll just mess the gel up. But I shouldn't mess up. Here we go. Let's add our loading die. Okay, let's double check everything. That's important. Loading die right there. Loading die. Now, loading die has glycerol and 2 dyes. It has glycerol, xylene cyanol, and bromophenol blue. Here we go. Talk about those in a second. Uh-oh. 1. You know what? I hate to waste tips, but I don't think this is going to work like this. I am going to change tips. I don't want to waste tips, but I'm going to have to. I just don't want to cross-contam. Here we go. Loading die in. New tip. Don't like to waste plastic if I don't have to. Loading die in. It's just because I don't want to get any DNA from the tube into my loading die. The great thing about loading die is that if I do mess up, I'll know it because it's so blue. This color is, at least. Some loading dyes are red. It's so blue that I'll know if I didn't put it in. I'm going to cap these up. And then give these a little tappy tap tap.
Mr. Jones?
Yes.
Just to clarify, like when you shine the UVC light, which one lights up, the loading gel or like the gel? I forgot the name already.
All right, before I answer that question, watch this magic. Ready? You see that's clear?
Yeah.
Ready? Wow. Oh, isn't that cool?
Yeah.
Let's see if we do that again. That one already did it. See, that's clear. Ready? Watch. Wow. Isn't that awesome? I'm just tapping it down like this, and the loading die is jumping to the bottom because it's so dense. So the loading die has glycerol, xylene cyanol, and bromophenol blue, and those 2 dyes plus the glycerol is more dense than DNA. That's why it's called loading die, because it's more dense than DNA and the salt solution that we're going to run the electricity through. So the loading die is going to get the DNA to sink. Okay, so who asked the question? I'm sorry, I can't see names because I have my screen so large. That is Tanha?
Ananya?
Ananya, I'm sorry. Go ahead, Ananya. One more time, please.
Which one lights up under the UVC light? Is it the loading dye or is it the gel?
It is the ethidium bromide in the gel.
Okay.
So the gel is inert with respect to fluorescence. It doesn't do anything. The gel is like a filter. That's all it is. It's a filter for DNA size. But the ethidium bromide does what's called intercalates. It intercalates into DNA, any DNA, even human DNA. It doesn't matter. That's why I'm wearing gloves. So if it intercalates in DNA, it gets stuck between the DNA. And then at some point, when I take the picture, when I shine UVC light through it, at some point, It's going to glow orange.
Okay. Thank you.
You're welcome. All right, here we go. This is a little boring. It's the same thing over and over again. And I hate to waste tips, but I don't want to cross-contam. This is period 9s. I'm just adding the same loading dye to everyone's tube. Those of you guys that are watching this video in the future, you should fast forward to this part. Because it's monotonous. But I'm just adding loading dye to everyone's tube over and over again. All right, let's do the magic show now. So once again, the magic show is where we close that up. Close my tips. Is where we cap all these tubes and then watch them go from blue to clear to blue. Ready? Wow. Nope. Wow. Yay, look at that. Yeah, I'll do that a little larger here. So that's clear. Yeah. No. I'm just shaking it. Come on. There we go. So how am I doing that? You can see it's clear, but there's a little blob of loading dye up here. And then I just shake it and the loading dye drops. Look at that, and turns blue. That tells me the loading dye, I can probably do them all at once. It tells me the loading dye is dropping down. There we go. See that? They're all blue. That's what you want. You want them all blue, or the color of your loading dye, so that the loading dye mixes with the DNA. Because we have to get this to sink into the gel. All right. These are all looking good. See, it's good to double check, triple check everything. Always double check your work so it's uniform. Okay, so period 1, period 8, period 9, 10. All right, now let's put these aside for a minute. Let's check our gels. Remember these? Remember these little boys? All right, so look. Look at that profile view. You can see the well of the gel cast by the comb. Nice and fat and very looking opaque. I'm feeling it. It's not that hot. So I think it's cool. All right. So it's time for surgery. So every gel box, every gel is different. Every gel apparatus is different. These have these black bumpers, and the bumpers, if I pull these black bumpers away, they're going to pull the gel out. See, I need the gel in this plastic case with the comb. So I have to get the gel off the black bumpers, and I use a ruler. And so I'm just going to perform some surgery on both sides. Normally you guys would do this. I'm performing surgery. I just like to say surgery. I'm performing surgery on this gel. You may go, Jones, well, you use the same ruler for both gels. It doesn't matter because there's no DNA in this gel yet. We're going to add the DNA. That's what we just prepared. Okay. We wash that off. Now, when I pull this black bumper away, it shouldn't pull the gel away. Let's see. And look, it didn't. Look at that. Isn't that beautiful? This feels like a cooking show. Look at that. Gel, tray, comb. I'm just putting those in a biohazard bin because I will clean those. That's that noise you hear. I'm throwing them into a biohazard bin. I will clean them because technically there's a ethidium on here. Now, it's not the bromide, it's the ethidium. Whoopsie, I missed. All right. So now let's put these in chambers. Let's see, what's the best way to see this? I'm thinking this. Like that. I think we'll go like this. I think we'll go like that. Okay, this is a little wobbly. Here. Okay, maybe that's better. That's probably better. Okay. Then I'm going to fill these up with buffer solution. Now this is TAE buffer. It's called tris acetate buffer. It's a weak acid, but it conducts electricity. That's the whole point. We're going to conduct electricity through the solutions. Clean as we go here. Now, I'm pushing the gel box in, the tray into the gel box, and look, the comb comes right out. I'll show you guys a view above in a second. Comb comes right out. And what we're left with is two gels with wells. That rhymes. Two gels with wells. There are 8 wells here and 10 wells here. 8 wells and 10 wells. So we're going to put the DNA into those little wells. And the electricity will run through these boxes. The electricity is going to run from the black to the red with the DNA. Well, actually, the electricity runs all throughout the field, but the DNA will run from the black to the red. DNA will run from the black to the red. The red is called the anode, and the black is called, excuse me, hold on a second. Let me rewind that. The red, as you guys know, is positive. So it exchanges anions. So yeah, so the red is the anode, and the black is negative. The black is actually the cathode. Now I know you guys go, wait a minute. Cations are positive. Anions are negative. But when you talk about cathodes and anodes, it's reversed, because anodes are positive because they exchange anions. That's an anode, and cathodes are negative because they exchange cations. So it's reversed when you talk about the odes, cathodes and anodes. Yes, cations are positive, and anions are negative. And DNA is an anion because DNA has phosphates. Remember the phosphates? So DNA is an anion. It goes to the anode. It migrates towards the anode, and that's the red. Let's load these gels. Let's find the best view for you guys. Let's see. I'm not sure the best view. I have to find the best view because once I find it, I can't move these again. I shouldn't. Maybe this is the best view. You know what? That's probably the best view, something like that. How do I do that? I'm going to prop it up like this. That might help. It's a little better. Let's do that. All right, here we go. So I'm going to get my little maps. It's the best view I can get, guys. So this is my little maps. This box is period 9. So excuse me. Yeah, period 9. You know what? I just realized I wrote period 3 on here. This is period 9. I'm going to put this like this. That's for me. And the picture later. And this is you guys. This is period 3. Excuse me, period 1. 1 through 6, period 1. All right. So I'm going to dial my pipetter all the way up as high as it goes, my P20. I'm going to dial my pipetter all the way up. 12, 13, 14, 15, 16, 17, 18, 19, 20. A little higher, actually. Don't tell. Here we go. So let's load the ladder first. I'm going to load, this is the 10 kilo base pair ladder. I know it says 1 kb on there, but that's because it's graded generally in 1,000 base pairs. I'll show you in a minute. New tip, I think you heard me get it. One second. Just adjusting. Just adjusting. New tip. I don't know if you can see it, but I'm leaning my elbow down to take the weight off my body. And I'm putting the ladder in the left-handmost lane. Get rid of that tip. New tip. And now let's do period 9's ladder. So I injected it, the DNA, into the well. It falls right in because it's more dense, thanks to the loading die, than the buffer solution. Now let's get a little interesting. I have another ladder I want to try with you guys. It's a 500 base pair ladder. You know, I figure since we're doing this, let's get a little experimental. This is a different ladder. I'm going to put all the way to the right. So we're going to use 2 ladders. I don't normally do that, but I figured I have the ladder. We're doing fewer gels. Let's use 2 ladders, and then whichever one we like the best, we'll use that one. Now, how do I know I loaded successfully? You can see the little blue dots in the gel. That means that the loading die is in the gel well. Let's load your samples. Now, guys, the bell is going to ring. I'm going to keep going. When the bell rings, feel free to take a break, whatever. I'm going to keep going because I don't want to lose my time. I'm going to keep loading these. So feel free to jump in, jump out when the bell rings. Here we go. Whoops. This is T1, P1, T1. super tiny. P1, T1. Loaded. That noise you hear is a new chip. Let's do P1, T2. P1, T2 is loaded. P1, T3. Loaded. P1, T4. Loaded. Uh-oh, what did I do? Why do I have 2 right here? 1, 2, 3, 4, 5, 6. Oh, I see what happened. 2, 1. Okay, I see what happened. No big deal. Okay. All right, you guys take a break if you need to. I'll be here doing this.
Mr. Jones,
Yes, sir.
I didn't want to interrupt you while you were working on the PCR gel, but I joined like 30 minutes ago. I had internet issues at the start of class though.
Okay. Do 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7. All right, hold on. Let me just finish this up, okay?
Yep.
Oh yeah, I'll talk to you later. That was Mr. Patel. Okay, here we go. That's T1. I can't move this box. Let's do T2. I'm sorry, period 9, excuse me. Period 9. One. You guys take a break. I'm gonna keep working. Oops. Seven. I think for the video, I'm gonna edit this part. Just speed it up. 8. Woo! and done. 8 plus 6 is what, 14? 14 loaded wells. All right. Power supply. Black to black, red to red. Black to black, red to red. Plug that in. Black to black, red to red. Will this reach? Black to black. Red to red. All right. Here we are. We are going. So this is the setup. So I have this on, let's see, we have till 9:04, a half hour. Can we do this in one? You know what? Let's run them on high. Maybe we can. 9:04. Can we do this in one? Maybe. So at this point, I'm going to pause the video. Before I pause it, though, just what's happening here is I'm running 120 volts from household current, household at the school, which is 110, 118, 120 volts, from this box through these electrophoretic fields. The electricity is running through these buffer solutions and therefore through the agarose gels. The gels are like big resistors, or actually they're pretty poor resistors. They're not big at all. Or I guess they are big, but they're just bad resistors. So the electricity is resisting through the gel and through the buffer solution, and it's pulling the DNA from the black to the red. DNA is negatively charged because of the phosphates. So it's going from the black to the red. The black is the cathode, the red's the anode. The anode is positive, the black is negative. The DNA is negative, it runs towards the positive. DNA runs towards the red, towards the anode, towards the positive charge because DNA is negative. Okay. One thing about DNA is that it's long and stringy. And one thing we're going to look at in the ladders in a minute, in the DNA ladders, is that DNA runs exponentially or logarithmically to the base pair length using base 10. So DNA doesn't run linearly. It actually runs logarithmically through a gel, meaning the smaller fragments run exponentially faster, than the larger fragments. The longer the DNA, the slower it runs, but not linearly. If you look at the DNA ladder, you'll notice that a 1,000 base pair fragment runs far slower than a 100 base pair fragment, and it's not linear. All right, so we'll pick that up later when we take the pictures. All right, I'm gonna pause the video because we're gonna sit here and watch these things, and we'll do notes while we do that. Stop recording.